Thank you very much, and thank you very much for the invitation to come and speak at this conference. I have no financial disclosures. You will see that there is 2 companies mentioned during the course of this presentation. We got data from them. We didn't get anything else from them. So I'm going to talk about genetic studies for reproductive aging. And I'm going to talk about the full span of reproductive aging when I talk about this. I'm going to start by talking about puberty timing a little bit, and then I'm going to move on to menopause timing. And towards the end, I might touch on some of the more behavioral timings that we've done some studies of. So I think everyone in this room is probably aware about why menopause timing is important, but I thought I might just introduce why puberty timing is also important. So this is a study done on about 250,000 women in the UK. And it was just looking to see what the observational, not genetic, the observational phenotypic association between puberty timing was and a range of diseases. And anything that's on this slide was significantly associated with puberty timing. So cancers, gynecological outcomes, musculoskeletal outcomes. There's also some psychological outcomes that come through there, respiratory outcomes, and general health-rated outcomes as well. One of the things that is potentially quite interesting here is that there's association with menopause, and that early menopause is associated both with early puberty timing and with late puberty timing. It's always good to be in the middle of the puberty timing distribution. And you'll see here early and late menarche. Early is broadly defined as between about 8 to 11. Late is roughly about 14 to 19. And the associations are increased risk of the majority of diseases in both cases. And the statistician in me is horrified by this plot, but it's a nice way of showing what I'm talking about here. So we have here the odds ratios of every single disease that we had outcome data for. So this is about 12,000 diseases, and we plotted the odds ratios. This is just a box and whisker plot of those. And you can see that actually across any disease you can think of, the majority of the effects is both in early and late. There's this U-shaped distribution. Being in the middle of puberty timing is good. And this is broadly consistent when we also look at puberty timing in boys. Far fewer associations here, a far more difficult to measure phenotype. So I work in puberty timing, and I couldn't tell you when my voice broke. So what we have here is rather than an exact age, we have earlier or later than your cohort. But it's the same sorts of things are coming through here. So some genetics, because this is a talk about GWAS. So this is a plot which is called a Manhattan plot. It's called a Manhattan plot because it's got a lot of things that stick up, much like the island of Manhattan. And effectively what it shows here is that we've got the physical positions across the whole of the DNA. All the various different chromosomes are laid out there, and how high they are is the strength of association that we have with the particular outcome of interest. In this case, it's menarche. So this comes from a study of over 300,000 women. GWAS studies are very large. They generally require a lot of data because the effect sizes are quite small in most cases, so we need a lot of data in order to find them. But we found 400 of them. These are robustly associated, correcting for a multiple test threshold because we're doing maybe 11 million tests across this, so we have to be very precise about which of those pass our multiple test threshold. And as I said, the individual effect sizes are normally quite small. Most of them are towards the end of that distribution, the 1 week rather than 1 year. And cumulatively, these top associations, these 400, explain about a quarter of the heritability. When I say heritability, what I mean is the amount that we generally ascribe to genetics, whether from twin studies or from other sorts of studies that try and capture all of the genetics at the same time. So what do we find here? Well, I'm aware that I'm talking to an audience where many of you may be clinical, so I thought I'd put this slide in just to demonstrate that we do occasionally find things that are clinically relevant, because 1 week may not mean anything to you from a clinical setting. And we found variants in this gene called TACR3. Now, we found a non-coding variant occurs in about 15% of the population, and this has an effect of about 2.5 weeks. Again, nothing clinical about that. But this same gene, if you're heterozygous for loss of function, about 1 in 1,000 people, then you've got an effect of about over a year. Again, maybe not the sort of thing where you'd actually turn up in any sort of hospital setting or clinic setting about that. But if you're homozygous for the loss of function, then you may have completely absent puberty. So the GWAS study may, in fact, reveal some specific biology which can then tell us about some more clinical outcomes, even though what we're looking at is the populational distribution, not necessarily the extreme pathological cases. So hopefully that's convinced you that we have at least some utility here. So the other thing that we can do is we can look at what other studies that have mapped the associations across the whole of the genome have told us. So this is a concept called genetic correlations, and what we do here is we take every single variant, every single SNP, it's called single nucleotide polymorphisms, and I may be using that towards the rest of this talk, and we look at the effect sizes on, in this case, menarche, and also the effect sizes for other outcomes that we may be interested in. So here, we can see that there's a strong genetic correlation with BMI, hip circumference, waist circumference, sort of anthropometric body shape type traits. There's a very close relationship between energy sensing and energy intake and puberty timing, especially in girls. But also we've got things like type 2 diabetes, fasting glucose, fasting insulin coming up as significantly associated. So this suggests that there are areas of the genome which have core implications for both metabolic disease and for puberty timing. In fact, this is replicating the phenotypic associations that we saw earlier. And some of these are flat. Bone mineral density, for example, puberty timing doesn't appear to have a huge amount of impact on that. So that's one of the things we can do once we've got genetic data. The other thing we can do, and this is a little bit more of an art, is we can try to link variants, SNPs, to biology. Now, this is often a problem in genetic studies because what we get out of a GWAS study is simply a physical location in DNA. That might be in a gene, it might be miles away from a gene, it might be something where we know about the effect of that variant, it might be where we know nothing about the effect of that variant. So what we end up doing often, and this is not necessarily to our credit, is we just go, what's the nearest gene? Now, if we look at one particular example, that might be a little bit too hasty, but if we're looking across 400 variants, then the best guess is that many of those will in fact be the most appropriate gene to choose. And once we have linked it to a gene, then we can start linking it to a biological understanding of the phenotype they're interested in. So again, here is puberty timing, and we see these sorts of genes coming up. So again, energy sensing, homeostasis comes up, FTO, a classic BMI variant, NEGRI1, another classic BMI variant. But we also see things which are more specific to puberty timing, GnHR secretion, pituitary development, and hormone synthesis and bioactivity. And also there's some peripheral feedback variants. I'm going to flag ESR1 because that's going to become important later on in the talk. So much of what puberty timing looks like from a genetic point of view is it's controlled neuronally, that many of the things are brain processes that spark puberty timing. But that may be a little bit hand-wavy for some of you in the audience. One thing that I'm just going to very quickly detour because it's kind of cool is this idea. So some of you may know about the concept of imprinting. So imprinting is where there is a different effect based on whether you inherit a variant from your mother or from your father, i.e. your mother or father may methylate various parts of the genome, and you may inherit that methylation, which may turn on or off the variant that you inherit. So if we look here, we've actually got various genes, and again, we're using gene names rather than SNP names, just out of convention from genetics, where if you inherit it either from your mother or your father, it has a strong effect, but it doesn't in the alternative parent. And slightly annoyingly, this is not consistently that you have paternal inheritance having an effect and maternal inheritance not having an effect. There's that slightly one in MEG9, which throws the whole pattern. It's a bit annoying, but there we go. But this is potentially something that's very interesting from an evolutionary point of view. Because if you think of a lot of what the evolutionary theory about parental investment tells us, is that there may be differences in the optimal strategy of males and females. Males may want children that extract the most resources they possibly can. Females may want more equitable distribution of resources among all of their children. So the fact that you can think of puberty as effectively the end point of childhood growth, the period when parental investment may be key, is not necessarily surprising that we see this most strongly. And of all the traits that people have measured, we see parental effects most strongly in puberty more than any other. So that was a slight detour, but I just thought that was potentially quite interesting. One thing that I find is quite important to emphasize when I'm talking to audiences who may be less familiar with GWASs is there's this concept that if you have a genetic variant, that is fixed. And in clinical genetics, where you have one highly penetrant, highly effective variant, that is often true. But this, because we're looking at a situation where we're looking at many common variants, all of small effects, there's a slightly different impact of what we're talking about here. So this was a study that we did in a Japanese population. And the reason I'm showing this is because there are differences in puberty timing between the Japanese population and our UK-based population. So this is what this histogram is showing here. So the European population is in blue, the Japanese are in red. And you can see that in general, Europeans have earlier puberty timing than the Japanese population. And the heritability estimates are also really quite different between them. The amount of the total genetic effect on puberty timing is different in European populations and in Japanese populations. It's far higher in European populations. And then when we look within the Japanese population, we can see that actually this is changing. Both the timing of puberty is changing and also the amount that genetic effects change puberty is changing. Genetic effects exist in an environment, and if you change the environment, you may well change the impact that genetics has. So sometimes we're looking at this and we're not thinking of providing an intervention that targets specifically the genetics. We may be looking at an intervention that changes the pathway through which the genetics acts. That's a key point for genetic studies. Okay, so all of the stuff I've talked to previously about has been in girls. Again, it's more memorable for girls when they went through puberty. It's less memorable for men. But a PhD student in our group has recently tried to take various sources of data. So this includes both voice breaking and timing of facial hair. And it includes both those where it's a continuous year measurement and those where it's just an early, normal, late measurement. And we think that there should be correlation between all of these measurements if we had true genetic variance. So by leveraging that expected correlation, he was able to combine all of these outcomes into one particular study, and we took appropriate corrections for doing that. So we end up with an effective sample size of about 200,000 men, and we find 76 signals. And 30 of those are novel in men compared to in women. And here is a list of the male-specific loci, which I've highlighted a few that might be particularly important. I'm going to particularly key into some of the fact that there's pigmentation variance in here. This is not something we expected. The idea that the neuronal control of pigmentation, because many pigmentation genes act primarily through cells which were originally neuronal, also have an effect on hair color. We found that potentially quite interesting. However, despite me saying that 30 of them hadn't previously been identified for puberty timing in women, there's actually quite a lot of coherence between what drives puberty in boys and what drives puberty in girls from a genetic point of view. There's quite good correlation. You'll notice there's a couple that stand out. MKRN3. Hopefully you remember that was one of the ones that had a paternal and influenced inheritance. So not only does it matter whether or not you inherit it from your mother or your father, but also the effect changes whether or not you're a boy or a girl. Also, SIM1. SIM1 has this very odd effect that it has divergent effects in boys and girls. There's a very strong effect to increase puberty in girls and a very strong effect to decrease puberty in boys. We don't understand that. If there's anyone in the audience who has an endocrine background who might be able to help us there, I'd be delighted if you came and talked to me. So that was puberty timing. So let's move on to the end of the fertility window, menopause. So this is a study which we did with 70,000 found 70,000 genome-wide samples. And we also did, in the blue here, a sample in a more rare set of variants. But we knew that they were coding variants. We called this an exome study. So rather than just looking across the whole of the genome, we focus in on those variants which we think are coding. And we had 40,000 people for those. And we identified 44 regions, so slightly fewer than we did for menarche, but it's a smaller sample, so we might expect that. But we also found 2 of these regions where we had a rare effect. This came through from our exome study. And as you can see from that purple line there, they were also represented by our common variant, our GWAS study. So what we find in common variants in the general population is mirrored by what we find in rare variants when we do those sorts of studies. Now, some of you may legitimately have thought that some of the stuff I was telling you about the biology of menarche was a little bit wishy-washy. Menopause is not that at all. Menopause came through with an incredibly clear message. 2/3 of the things that we identify were genes that we knew to be involved in DNA damage response. And we didn't actually have to do any fancy pathway analysis for these sorts of things. We could just look at them. There was BRCA1, there was BRCA2, there was HELB, classic DNA repair variants. So we think that what the genetics is telling us about menopause, certainly from a genetic point of view, is the key thing for the timing of menopause is the ability of oocytes to detect and repair DNA damage. It's the loss of oocytes because of DNA damage that's the major driver of the timing of menopause. However, it's not necessarily quite so simple. Now, this is a mechanism which is unambiguously DNA damage, but it is telling us something slightly different. So this is the example of CHEK2. Now, for most of the DNA damages, genes, if you have mutations that affect your ability to detect DNA damage, you will have earlier menopause. Your oocytes are less viable. But the CHEK2 story is a little bit different. So in CHEK2, those people who have a functional CHEK2 induce P63, P53 induced apoptosis in oocytes which the body determines to be non-viable. But those people who have not got a functional copy of CHEK2 actually have later menopause because the body's error detection is working less well. So these people will present with menopause that is much later, but their oocytes may be of lower quality, and that may have an effect on their fertility as well. And this is something that we're investigating further, but unfortunately I don't have anything particularly concrete to tell you about that, I'm afraid. And the one thing that we can also do is we can start looking across the various different phenotypes. And we can see that there are some core genes that turn up in both cases. FSHB, for example. FSHB looks like it's shifting the reproductive window across the board in all areas. It also, this variant, increases your likelihood of having twins. So it looks like there's a certain element of just speeding up reproduction in all cases. But we also see common and rare variants and across menarche and menopause. So there's a core set of variants which are involved in reproductive timing in all areas. Okay, so you may have been a little bit surprised that I was a little bit down on the biology and what GWAS can tell us about biology, because there's actually some epidemiology that GWAS can tell us about, and I think potentially this is a lot more exciting. And I'm trying to act as a bit of an evangelist here and tell you about what GWAS can do for you. So those of you who are familiar with epidemiology will know what this sort of diagram looks like. We've got an exposure and an outcome, and we would like to know what the association is, but we're worried about confounders. And confounders are a constant problem. We can measure them, we can control for them, but some of them are unmeasured. So we in genetics have a slight sleight of hand. It's called Mendelian randomization, and this is where we use SNPs as a proxy measure for any exposure that we might have of interest. And you'll see that there are, deliberately, because I drew this diagram, no lines between SNPs and confounders. This means that the SNPs aren't associated with our confounders. And this means that we can, if we do use them as a proxy, get an unbiased estimate for the effect of the exposure on the outcome. Now, some of you may be thinking, how can this possibly be true? But actually, when we do most of our studies, we find that the GWASs are specific to the exposure that we're interested in. And actually, this holds in the vast majority of cases, that we can use SNPs as a proxy without necessarily having to worry for confounders. And there's no reverse causality here. There are very few outcomes which change the blood DNA that we extract from someone. It's not like there's major changes going on there. So we know that the SNPs have come first, and therefore we're impacting the exposure. So that's the logic that we use, and this is the equations that we use. Yeah, hopefully we assume that the blue line is true, and then we can forget about everything else. So I'm going to come back a little bit to that thing I had about male puberty timing and hair color. So there's a robust phenotypic association. People with darker hair are more likely to have early voice breaking. And this is consistent across most hair colors. And this is also seen phenotypically in girls as well. Girls with darker hair likely to go through puberty earlier. But puberty timing may have associations with your ancestry group. And that may have an association with the socioeconomic status. And that may have an association with when you go through puberty. So when we do the genetics and we look to see what, if we group together all of the variants that are associated with hair color, do they have an effect on puberty timing? And they do in boys, and they don't in girls, which kind of mirrors what we saw. We didn't find any of those puberty timing SNPs variants when we did the initial study. So there does seem to be this specific association with neurological control of pigmentation that also affects puberty timing in boys. Also affects your lifespan, i.e., if we look at people who are older, who have lived longer, and we look to see whether they are enriched with puberty timing variants that either increase or decrease your timing of puberty, we can see that people who go through puberty later live longer, or rather that more people who have lived longer are overrepresented in genes that cause them to have had puberty later, and this is true of both men and women. So there seems to be something that's associated with puberty timing earlier that affects lifespan. And if we go back to those original forest plots, we can probably guess that some of those diseases that puberty timing is associated with might be killing people. So just to very quickly talk you through this one example, and this is potentially quite complicated. So we wanted to know about the association between puberty timing and breast cancer, because we think puberty timing has an important impact on hormonal exposure in the life course, and we thought breast cancer would be a good example of this. So, and then we did it, and it was flat, which was somewhat disappointing. But we then remembered that a number of our variants were also associated with BMI. They might actually have an actual arrow that links the SNPs, the variants, to those confounders. So then we split our variants into two groups. We said some of them were associated with BMI and some of them show no association with BMI. And we're going to do that experiment again. And when we took into account BMI, there's this weird association, which I don't think anyone has explained yet, that increased BMI increases your risk of breast cancer. It doesn't match any of the phenotypic associations whatsoever. This is something that people are looking very closely. But reassuringly, because we were interested in puberty timing, those that decreased your timing of puberty timing did increase your risk of breast cancer. So these are people who have later, and they have a lower odds ratio, lifetime exposure to hormones. And this is a pattern that we see across other reproductive cancers: breast cancer, endometrial cancer, prostate cancer. Now, for those of you who are paying close attention, you'll have noticed that I've just suggested that people who go through later menarche have a lower risk of prostate cancer. This is because, as I showed earlier, the genes and the SNPs that underlie puberty timing are common between boys and girls. So actually, we're measuring very similar things when we have a genetic score for either puberty timing or in boys or girls. All right, so this is my final couple of slides. And I thought I'd just talk about a more behavioral impact that we have for genetics. So this is the age at first sexual intercourse, and we did a study of this. And we estimate the genetic effect on this is about 30%. And we found 38 genetic loci that are robustly associated with this. Now, potentially unsurprisingly, these loci have also, some of them, been associated with puberty timing. Those people who develop sexual characteristics earlier start having sex earlier. Makes sense. But some of them have influences that we think may be more personality-driven. The classic one is a CADM2 region, which is also associated with risk-taking behavior very broadly defined. So this also makes you smoke earlier in life. It makes you more likely to speed on the motorway. It has a whole variety of associations that look like they're associated with risk-taking. I quite like talking about this one because it means I can show this slide. So as you can imagine, this study generated a certain amount of media attention. I like the fact that this is the Sun, by the way, for people who know about English newspapers. If you're German, this is like the Bild. If you're American, this is like the National Enquirer or the New York Post. It's the only time they've ever written about my research. And they obviously thought that they had two different headlines and couldn't decide which one to go with. But, and the Sun is not famed for its quality of its scientific report, they did actually get one of our key messages here, that sex at an early age is linked to poor school performance, ill health, and psychiatric problems. Now, why were they saying that? Because we'd had this graph in the paper. This was a graph, again, of these genetic correlations. What else shared an underlying genetic biology with it? You'll see here, years of education, very, very strong effect. The same things that are driving people to have sex earlier often decrease their performance in schools. Also, age of first birth, totally understandably. People who start having sex later also start having children later. And you can see also number of children. People who start having sex later have fewer children. Again, not particularly surprising, but this is just demonstrating that you can use genetics to try and give further evidence for phenotypic associations. And this, I think, is where a couple of people often go, but you're talking about a genetic influence on behavior. Why would you do this? Years of education. Now, years of education is really quite important. And if we go back to the Japanese example, we know that genetic effects only operate through particular environments. So if the structure, the teaching ideas, that exist in education are actively dissuading people who are having earlier sexual activity or earlier disruptive behavior in some way, we can change that. We can mean that those people who at the moment may be doing less well in education, we can start identifying some of the reasons why that is happening, which might be related to their reproductive behavior, and potentially try and understand if there's a way that we can accommodate these people a little bit more. All right, so one of the variants that came out of that study was ESR1. Now, ESR1 obviously has a peripheral effect, but I think this was the first time that we'd ever demonstrated that a hormone had a strong effect on a reproductive behavior. And this supports the idea that there's a neurobiological role for ESR1 in both men and women. It was very strong for both of them. And to come back to some of the work that one of our PhD students did right in the last slide, ESR1 is a really interesting region of the genome for reproductive behavior. So this is some work he did where we basically looked at every single reproductive outcome we had data on, and was it associated at all with variants of the ESR1 region. And two things stand out for me here. First is, yes, there's a lot of associations here across a whole range of different outcomes. And the second is that, unlike usually in genetics, if we see one variant that's associated with, say, BMI and puberty timing and breast cancer, it's the same variant in all cases. The signals line up very nicely. That's not what's going on here. There's very specific associations associated with very specific outcomes. Now, this might be to do with tissue-specific expression, epigenetic control of those sorts of things, but it's certainly something that I think warrants further assessment. Okay, so in conclusion, population studies have been very successful at identifying many regions of the genome associated with reproductive timing and indeed function. Some of these outcomes are very specific to the outcome that we're looking at. Others have much more widespread effects. And we're beginning, though I would be the first to admit we're not there yet, to understand how these might have biological messages that we can take forward. But even without those biological understanding, we can still use this in epidemiological settings to understand the relationships between reproductive function, later life diseases, and even socioeconomic outcomes in some cases as well. And overall, the downstream consequences of variation in reproductive timing has important impacts across a whole spectrum of features in life. And we are, of course, doing larger studies to try and find out more about this. So if you have interest in joining those sorts of things, do come and talk to me. Thank you very much.
Thank you. Thank you very much, Felix, also for keeping the time. There will be room for a few questions. I think the GWAS is an eye-opener to many of us, but at least when I heard your colleague, Perry, speak about it, I saw that in the future we will get to much more about it and also the clinical application. But if you have questions for Felix, please come forward. We may have also, by the end of this session, a possibility of having questions where we can maybe link the different presentations together. So,
I'll also be around afterwards if people would prefer to ask me questions in private. So that's something they would prefer.
Thank you once again, Felix.
Thank you very much.
So I think I was inspired by listening to your colleague when I tried to set up also my own lecture of today. I am from Copenhagen, Svend Skovby, and I'm a clinician. I'm working more with reproductive medicine. But here are my disclosures. I think for this lecture, there are no conflicts of interest. But I felt fascinated by the GWAS and with the upcoming information on translational medicine in order to look at also some reproductive issues, especially the reproductive aging in females. So translational medicine is defined as an interdisciplinary branch of the biomedical field supported by three main pillars: bedside, and community. And the goal is to combine discipline, resources, expertise, and techniques within these pillar is to promote enhancement in prevention, diagnostics, and therapies. But how and why should we apply translational medicine to reproductive aging? Well, I think it's important because translational medicine may disentangle how the physiology and endocrinology of female reproductive aging interplay with the genetic, epigenetic regulation and functional genomics of the aging process. So here is a cartoon on normal, basic biological aging and factors that will finally end up with what we can call physiological neuroscience. And it gives us information on functional changes, mechanisms behind prevention, treatment strategies, and so forth. And maybe we could pinpoint out in reproductive aging items where we could be allowed to have a deeper insight in the mechanism and maybe also for intervention. So the perspectives of applying translational medicine could be more insight, as mentioned, in variation in onset of time of menopause, variation in severity of symptoms, and maybe also enable personalized medicine by use of pharmacogenomics when we are offering menopausal hormonal therapy. Wishful thinking, at least. First here, it is a fact that age of menopause, or not to a higher degree, but does vary according to where you are looking on the global area. And this, of course, will indicate that there are environmental, but also maybe genetic, epigenetic factors that are interplaying and finally result in these differences in the onset of menopause. And furthermore, if you look at in the women, the age when sterility may take place, there's a great variation in the age ranges. That's number one here. Number two is sterility. Number three is onset of irregular bleeding. And number four is, again, menopause. So this variation in the age for these phenomena to occur could be explained maybe by genetics, epigenetics, but obviously also with some environmental factor that is, of course, also linked to epigenetics. But if we go into the physiology and endocrinology of female reproductive aging, it's fascinating, if we look at the physiology, that in the female fetus, already when the fetus is around 8 weeks, we will see that there's the highest number of oogonias, stem cells for oocytes, because a fantastic mitotic process has taken place, and thereafter the first meiotic cleavage will be starting, and in a given follicle it will end up only 50 years after. So it's a very special situation that if you look here at the timeline, you have oogenesis caused by mitosis, creating the highest number of oocytes before the 6 months of pregnancy, and thereafter it stops, and you have folliculogenesis going on. But you will also see that atresia takes place. So there is a constant atretic background for the decrease in number of oocytes. So at term, there will only remain 1 million, and when a woman gets fertile at the age of puberty, only 3,000 follicles remain for the period of reproductive age. I will dwell a little bit on the normal physiology. You will see here the development from primordial to pre-ovulatory follicle size, the GWAS follicle. And the whole process takes around 200 days, 180 days. And you can see here that the development from small antral to pre-ovulatory follicle is depending on some interfollicular mechanisms and also some endocrine signals. So we have AMH as maybe limiting the number of follicles that enter each cycle. We have, when the follicle has a certain size, approximately 6 millimeter, we have receptors in the granulosa cells for FSH. So thereafter, the maturation process is conducted via FSH stimulation. There are much more involved here, activation systems, and we have no time to go into this. But it could be of interest maybe to find out if there is genetic, epigenetic interference in the maturation process, in the velocity of atresia, and in the selection of the single follicle that will be ovulated in a given cycle. Before going into such a discussion, there's one more thing that we may need to take into consideration if we are looking, again, basically on female aging, and that is the release of hormones from the pituitary hypothalamic system, where we have a close connection between the neurotransmitters from the serotonin system, from the dopaminergic system, and from the noradrenergic systems, which in fact are secreted together with the GnRH and will determine also the pulsatil decrease or release of the anterior pituitary hormones, FSH and LH. Going back here, you will see again in an overview the travel from primordial, or the development from primordial to the GWAS follicle. And again here, it's evident that in any of the different phases, atresia can take place and will take place. It's also obvious that the process with atresia will accelerate according to the chronological age of a given woman. So atresia is much more common in the age between 40 and 50 compared to the beginning of the fertile period. And once we have reached a number of approximately 1,000 follicles, menopause will take place. There only remain maybe 1,000 follicles, not enough to produce estrogen to create bleedings anymore. So there has been an aging process. There has been atresia that leads up to this situation. Of course, there are variation, a normal statistical variation between one woman to the other on the number of follicles that exist at a given age. But in general, we will see that around the age of 50, there will only be 1,000 left, and that will lead up to menopause. We have some idea on how close a woman will be to menopause by using a paraclinical variable, and that's the AMH, as we saw, was secreted from the granulosa cells in the small follicles. So we are able of measuring, in fact, how many follicles, or have an estimate of how many follicles are left in a given woman at a given chronological age. But we will see that there is, again, quite a variation in the levels of AMH according to the chronological age, and that will also impact the onset of menopause accordingly. So, if a woman has an early menopause, she will suffer from different comorbidity. And it is a fact that bleedings will be irregular already at the age of 40, indicating advanced aging of the follicular system. And we also know that in some women, not many, but in some women, we will see the last bleeding before the age of 40, what we describe as primary ovarian insufficiency. It may be existing in 1% to 2% of all women. And these women will, as mentioned, have comorbidity. It will have long-term health consequences in relation to coronary heart disease, dementia, Parkinsonism, osteoporosis, and other important, conditions for having a normal aging process. So if we, apart from measuring AMH, could determine those women who were at the highest risk of having early menopause, we will probably be able, maybe not tomorrow, but on the long term, to intervene. Now, we have trying to go into the aging process by looking at granulosa cells to find out if there is a distinctive epigenetic aging profile. And by looking at that, would it be possible to pinpoint out those women who have the highest risk of an early menopause? So the research question has been, does women's age affect the DNA methylation profile differently in mural granulosa cells compared to other somatic cells? So using DNA methylation as a proxy for the aging process, would there be special mechanisms in the granulosa cells which are supplying the follicle and the oocyte with nutrition and other agents that will secure natural follicular maturation? This is a busy slide here, but what it shows here that if you apply what is called a clock, epigenetic clock, which has been well used for many purposes, where you can look at different organs and find out by using the clock whether a given person has a natural function according to her or his chronological age. And you will often see that there is good correlation by using this clock and compare it to the chronological age. But this slide here indicates that if you look at leukocytes from peripheral blood and compare the situation with applying the clock on mural granulosa cells, you will see that they have a much younger age compared to leukocytes when again comparing to chronological age. So it seems that granulosa cells appear younger compared to other organs in the body. But if you look at the correlation between leukocytes and granulosas in a given individual, there's a good correlation. But it seems that the traditional clock system are not sufficient to measure the age of granulosa cells compared to other cells. And also, if you look at age acceleration in the leukocytes representing normal somatic cells, you will see here, if you compare on the left-hand side, that there is an acceleration according to age, whereas this is totally different if you look at mural granulosa cells. And that is also what is translated from the right hand of the slide, that there's a great difference if we look at granulosa cells with a clock system and the somatic cells. So we tried to work out a clock system that were more precise when looking at granulosa cells to find out in a given woman if she was deviating from another woman in order to predict a kind of early aging in her situation. We applied also the clock system on leukocytes and found here it was possible, but the age of the leukocytes were highly overestimated with the granulosa cell clock. But the granulosa cell clock is a kind of possibility in a given woman to find out if she has an accelerated process in her follicular aging compared to other women. So eventually you could compare AMH with the age determined by the granulosa cell clock and predict more precisely when menopause would take place or the fertility potential in this woman. We also looked at telomere lengths and found out that the telomere lengths, in contrast to looking at other cells, other organs, did not in granulosa cells indicated anything about the aging process. So we, in short, found out that it was possible to predict or point out age predictors, the DNA methylation age in granulosa cells, and it was found considerably younger compared to the chronological age. And we could see that the clock was predictable of the age. It was also predictable in leukocytes, but here we found a higher age compared to other clock systems. Another finding was that the mural granulosa cells differed from leukocytes in having a higher number of epimutations, which may be indicating that we have a chance of having genetic problems or the genetic material will differ according to the age of the granulosa cells or to the age of the woman. But obviously, what we heard before, the GWAS has much more possibilities compared to just looking at the epigenetic aging process. So I will not go into detail because we had such a brilliant lecture before this one here, but just to indicate that the GWAS will allow us to have a good insight in the genetic disposition that may lead up to both reproductive failures, but also to important issues in relation to diseases and BMI. And here just an indication of some genes that are more expressed, more common in women who have an early menopause. Felix can tell you much more about that. But going back to the long period of development from the female fetal state and until menopause, it's evident that if you have the follicle, the oocytes, arrested in the first meiotic division, there is a good reason that there may be impact or failures or DNA damage in that period. So it makes or underscores the importance of applying GWAS examinations if we would like to have the optimal reproductive situation and also to be void of later comorbidity in the women that are coming into a pregnancy after the release of the oocyte and fertilization in the tube. So maybe you have said, Felix, but the genetic etiology of early menopause and primary ovarian insufficiency is largely unknown in the majority of cases. And look again, it's only 1 to 2% of the whole female population. But the GWAS has discovered variants previously associated with normal age at normal menopause were also associated with early menopause and primary ovarian insufficiency. So we have increased our knowledge on when and why primary ovarian insufficiency can take place. And early menopause has a genetic etiology which overlaps variation in normal age at menopause, but it at least partly explained by some additive effects on the polygenic variants. And the combined effect of common variants captured by single nucleotide polymorphism arrays was estimated to account for approximately 30% of variants in menopausal age or in the occurrence of early menopause. So again, indicating the genetic assistance we can have to be more precise in the prediction of early menopause and in women who have an accelerated aging process in the ovaries. And now, finally, there's another translation by using genetics, and that's to pharmacogenomics. Because together or parallel with GWAS, there has been made progress in understanding the influence of genetic variation on estrogen efficacy and metabolism. So the goal of pharmacogenomics is to use genetic information to predict how an individual will respond to a drug with the ultimate objective of aiding clinicians in selecting the right drug in the right dose at the right time for every patient in order to ensure drug efficacy. And see, here you will just have an indication that according to given genotype, you will have different effects from applying post-menopausal hormonal therapy or any other kind of drug hormonal therapy. So we will be able to find out what is the therapeutic window, what may end up in toxicity, in which women will have no efficacy based upon the genetic situation. So I will just end up by concluding that several factors, including the women's age, the reproductive age, baseline vascular health, biomarker levels, genetic and epigenetic predisposition, appear to modulate long-term female health, but translational medicine, including pharmacogenomics, enables the optimal personalized approach to reproductive medicine and also to menopausal hormonal therapy. Thank you for your attention. Have we time for 1 or 2 questions? Yes. If not, it's my pleasure to invite last speaker to the rostrum.
Können wir gemeinsam Ihre Präsentation finden?
Michael, you are here from Austria. Maybe
yes.
You will give your academic background yourself.
Ladies and gentlemen, I would also like to thank the organizers for the invitation to come here. I'm just like Sven, I'm a clinician as well. I did my training at the Medical University Clinic of Vienna, and I'm working as a postdoc at Karolinska Institutet in Stockholm as well. And I will talk a little bit more at the possible clinical application of what you just heard. We have it everywhere in the Western world that women are advancing their child wish. And this is data from Austria from the 90s to 2010. The age at first birth was rising from the mid-20s around the 30s. And I think we have it everywhere in the Western world. This is a beautiful study from a scientist, Eva Bourgeois, who is working in Austria as well, who looked at the birth years from 1950 to the 1980s or to 1980, and looked at if women are childless and are wishing to have a child in the age between 35 and 39. And what you can see here is that this proportion of women between 35 to 39 is steeply rising. And as well, as you can see in the green dots, those women who stay childless, even though they want to have a child, is also dramatically rising in the Western world. And this paper from her group as well was just published about a month ago in Human Reproduction, shows that women around 30 years, this is Austrian data as well, 70% want to found family and want to have children. And the same women report a couple of years later that only 20% of them want to realize their reproductive intentions. So during these 10 years, these intentions are dramatically decreasing, and this has, of course, also to do with reproductive aging. And we heard a lot in this session about reproductive aging, and we have a certain number of women who have their so-called physiological ovarian senescence. We have a certain number of women who are affected by premature ovarian aging, and then we have those women who have premature ovarian failure. And as you just heard in the previous talk, everything above the age of 40 is thought of being a normal menopause. But a woman who comes to menopause with 41, 42, is experiencing premature ovarian aging exactly in this period where she maybe has an intention to found family between 35 and 39. So actually, this group that has a child wish and is close to the end of her reproductive lifespan is dramatically increasing because we are just delaying family planning. So as you already heard, premature ovarian failure is the menopause before the age of 40 years. It is thought to affect 1 to 2% of women. We have low AMH and high FSH, and the only solution for those women is often oocyte donation. But we have this big group of women with diminished ovarian reserve that we as clinicians see on a daily basis in reproductive medicine clinics. And the transition to menopause is a long process. So this woman who is getting to menopause, as I just said, with 41, 42 years here, might experience a reduction in her ovarian function already 5 to 10 years before that. We in our daily practice see about 1/3 of couples being affected by diminished ovarian reserve. These couples or these women have a reduced response to ovarian stimulation, have a reduced probability of getting pregnant in a natural way. They have an increased miscarriage rate, reduced embryo quality, and we as physicians have difficulties to treat those women. And the classical view on premature ovarian insufficiency or failure was that there is different environmental factors, immunological diseases, metabolic syndrome, whatever, and then about 20% genetic causes. However, the majority of cases of premature ovarian insufficiency remains undiagnosed. And as you just saw in the previous talks of Felix and Sven, the genetic causes, or let's put it like this, we learn much more and more about genetic causes of reduced ovarian function. And these genetic causes you can measure in quite a high number of patients with premature ovarian insufficiency and diminished ovarian reserve. And one of the latest reviews actually describe premature ovarian insufficiency rather than a disease that is multifactorial with a small genetic factor. These authors said this supports the notion that premature ovarian insufficiency is a heterogenic genetic condition that involves some other factors. So the latest research was showing that the genetic factor might be much bigger than we thought. And we know a lot of mutations that might affect ovarian function on different levels. So on ovary formation at the very beginning, then of follicular activation, follicular growth, oocyte maturation, and ovulation. So we really know where these mutations are affecting ovarian health. One other thing, when we talk about the clinics of ovarian aging, is that we nowadays have the possibility to freeze eggs of young women so that in a later age they can use them to prevent them to get infertile just because they delayed their child wish to the late 30s. And unfortunately, the usage rates are very low. So we have around usage rates between 5 to 10%. The costs are quite high. And there is certain countries, including Austria, where you are today, where the legal situation is actually prohibiting social freezing for non-medical reasons. So we can only freeze oocytes if there is some medical condition. And this review puts it quite nicely, the dilemma of social oocyte freezing. The usage rate, as I already said, is between 3 and 10%, and the costs per extra live birth is $600,000 to $1 million. So this makes it far from being cost efficient. And as you already heard, it would be, of course, a nice thing to put this aspect of premature ovarian aging and also prevention of premature ovarian aging in a personalized aspect. And especially my previous speaker has talked a little bit about personalized medicine. And this could make actually, for example, social freezing a fertility preservation technique in those women who really need it, in those women who might be affected by premature ovarian aging and who might therefore already much earlier get into diminished ovarian reserve or premature menopause. And you saw a couple of articles from The Sun, for example. Also the Huffington Post was getting up this topic, your genetics might affect fertility more than you think. And there is already, of course, a couple of companies that jumped on it. For example, the University of Chicago has a premature ovarian failure panel where they look at a couple of mutations that you already heard of today. There's a couple of other companies, Prevention Genetics, premature ovarian failure panels from Blueprint Genetics. I just Googled it, to be honest. I don't know these companies. And this review already showed up a couple of times. You have Felix here as a co-author. And as you can see, a lot of mutations might have an impact on ovarian aging, but the impact is not very high. As Felix already put it, 1.5 months, 2 months. From a clinical point of view, 1.5 to 2 months don't interest me at all. So why test them? And you have some rare mutations that have a high effect, and then you have some very common mutations that have only a very small effect. So that as a take-home message, and I will come back to that later, we should for sure focus on those ones, those rare ones that have a high effect, because no patient gets any benefit if I find a mutation that I find with anyways 50% of patients I test and might have no impact. We also designed a panel at our institute with around 120 mutations, and I want to point out some of the mutations we test to make a prognosis for premature ovarian failure or diminished ovarian reserve, which is Fragile X, BMP15, FSH receptor, and NOBOX. Fragile X, for sure most of you have heard of it before, is quite common in women with premature ovarian insufficiency. We have 3 to 15%, depending on the publications. And in 20 to 30%, on the other hand, of women with increased repeats, you find premature ovarian insufficiency. And especially those women with pre-mutations, so those who do not have the very long repeats and will anyways end up in premature ovarian insufficiency, there is a curvilinear association of the age of menopause. So depending on the fMRI repeats, we can predict if this woman is at risk developing an earlier menopause or not. However, in the normal range, fMRI repeats do not affect the risk of early menopause, so only in the pre-mutations. The FSH receptor is a mutation that was studied by a lot of published studies, and is, of course, as you already heard, a very important gene for oocyte and follicular maturation, for example. And there is a large number of publications, as I already said. This one is particularly interesting. It looked at FSH receptor in pre or peripubertal girls, and already in these girls, they could find FSH, LH, and E2 levels that were associated with mutations in FSHR and FSHB, and also the number of large follicles in these very young girls was associated with mutations in FSHR and FSHB. Similar study here. FSHB mutations were associated with higher FSH and LH levels and with idiopathic infertility in those women who then intended to have a child. And some certain combinations of FSHR and FSHB might affect AMH levels and FSH levels, as you see here, with women having significantly higher FSH and lower AMH levels. And this is another study looking at some similar measurements, and you see here, depending on the genotype, women had between 1.36 and 6.8 AMH. So I would say this is a clinical relevant difference. And I will not go in detail to that. There is a lot of studies investigating FSHR receptors on IVF outcome and response to ovarian stimulation. BMP15 is another protein that is important for follicular maturations, and mutations of BMP15 have been associated with premature ovarian insufficiency, and around 1.5 to 12% of women with POI have a mutation in BMP15. And as you can see, in this study, BMP15 was significantly associated with ages at menopause as well. And just as a last one for the examples, I will point out NOBOX is as well a mutation that was associated with premature ovarian insufficiency, as shown by different studies. If any of you would like to go deeper in this field, there is another nice review that pointed out a couple of polymorphisms and mutations. In our panel, we concentrated on mutations rather than polymorphisms because they're much easier to interpret than the polymorphisms. So we have several known mutations and polymorphisms associated with premature ovarian insufficiency or failure, with advanced reproductive aging, and with diminished ovarian reserve. And theoretically, we can prevent early reproductive aging and infertility to reduced ovarian function on different levels. And the treatment strategies depend on what mutations we find. So on those women with still active ovarian function, we might do ovarian stimulation and oocyte cryopreservation. In some women, and those of you who already have been here at Professor Pellissier's talk, he showed that women with premature ovarian insufficiency to a quite high amount, I think he said 30%, actually have follicles in their ovary. So ovarian tissue cryopreservation and extracorporeal activation might be a treatment option for those women. Advancing family planning is always easy to say and difficult to make. And oocyte donation in those cases where these treatment strategies come too late. And there is a very, very nice review that was published quite recently. And this review actually, in those women who already suffer from premature ovarian aging, suggests genetic testing and tests specifically for those genes involved with follicular growth and function. And those that still, or that have, for example, a mutation that affects follicular maturation might undergo fertility preservation and in vitro activation in the future. So we can also in those women who are already affected by premature ovarian aging and menopause, depending on our testing, personalized, make a treatment for them. Of course, the studies are very heterogeneous, and I already said when we talk about polymorphisms, you find a large number of studies that could show an association, maybe a clinically very small association, then a couple of other studies that show completely different. So we should only use mutations with a good evidence. And the exact clinical impact of many of those mutations is still unknown. The costs are getting lower and lower, but of course it's still not a very cheap procedure, so to say. So to conclude, we are still at the beginning. Big data, as you saw with the presentation of Felix, will reveal much more. However, again, we should concentrate on the clinical meaningful mutations when it comes to treating patients. And the tests will get cheaper and cheaper, and I think these kind of tests definitely will come to daily clinical practice to help our patients fulfill their child wish in their future when they have a partner and when the ideal time has come to found a family. Thank you very much.